So in this video, we have to talk about the regulation of the prokaryotic replication. If you have not seen that video, you can just have a view on that video, and after that you can come on to this video. Here we have to discuss about the regulation of prokaryotic replication, then we'll talk about the replication in plasmids, then the mitochondrial replication, and then finally the eukaryotic replication. So all these topics are short, then we'll move on to the eukaryotic replication. So if I talk about the regulation of prokaryotic replication, so here some things are very important. Now when I was talking about the replication process in the prokaryotes, uh, in the previous video, I told you this, that in the ori region or in the ori sequence, there is a particular what do we say it, GATC sequence. And this GATC sequence here, this A region and this adenine has to be methylated in that GATC sequence. If this adenine is methylated, then only the replication can begin. So one of the major regulation points come here, that if whole of the ori sequence is methylated, fully methylated, means the adenine region is methylated, then the replication can begin. Now this is done by the enzyme which is called as dimethyltransferase. Now this dimethyltransferase actually is doing nothing but catalyzing the methylation of the adenine in this GATC sequence. So until and unless the full methylation in the ori sequence does not take place, the replication cannot begin. Second important thing is that in this same ori sequence there is one 9-mer repeat. What is the meaning of mers? Mers means the base pairs. So there is a repeat of 9 base pairs, and in these 9 base pairs we have one very special protein which is DNA AATP. So what happens is the DNA AATP will do the DNA AATP. This particular protein will, at the 9 mers, it will do the melting. So as I was talking about the melting also, which is done by the DNA helicase and all, so the first melting, the melting even prior to what DNA helicase is doing, is that done by the DNA AATP protein. So it basically do the melting at this 9 mers region, and once it do the melting, it becomes DNA AADP. So if the DNA AADP is present, that means now the replication will not reinitiate. But if by chance from any other source, or even this, if again changed into DNA AATP, it can redo the melting at that position and it will restart the replication. But we do not want that. We do not want the DNA ATP to restart the melting. So for this there is one important protein which is called as HDA protein. The HDA protein will do the regulation. It will check for this DNA AATP, and if it is present, then it will convert this into DNA AADP. Means the unnecessary DNA AATP will be converted into DNA AADP by this HDA protein. But still, if by some cases this DNA AATP remains, this is the risky matter. So in that condition, that 9-mer sequence which is present in the ori site, it will be masked by a protein which is known as sequence A protein. This sequence A protein will mask this 9-mers region, and hence the DNA AATP will not be able to bind to this particular region. So this is the prokaryotic replication regulation. Now we'll move on to the replication of plasmids. So the replication of plasmids is also very much similar. In plasmids, if I talk about, the replication is very similar to what the replication was happening here in the prokaryotes. But here, after the melting, that is after the initial melting, there has to be a nick created. So here, firstly, I can say that the melting is done, and after the melting is done, there is a nick created. And this nick creation is done by certain proteins called rep proteins. Now when this rep proteins will create this nick, then only the replication will begin. So this creating of nick is very important. Now you should remember the difference between nick and gap. When I talk about the two nucleotides and their bonds, we know that this is 3' bond and this is the 5' bond, and they are linked together. But if by some case here this 3' bond and the 5' bond, they are not linked together, that time we say that this is the nick created, and this can easily be sealed. But if I talk about a gap, that means the absence of a nucleotide. So if this whole nucleotide is not present, that is what we call as a gap. So this nick is created by these wrap proteins in the plasmid at the ori region, so that the replication can initiate. Also we have to remember that here the replication takes place unidirectionally, unlike in the bacteria where it was bidirectional. And also here the replication is very high, means replication has to again and again happen, so that the copy number of the plasmids can increase, because that is what their main aim is. So for that, the regulation of replication here does not take place. There is no such regulation of replication happening in plasmids. So these are the 2-3 important points you need to learn about the replication in the plasmids. Now I will talk about the replication in mitochondria. How does the mitochondria replicates? So let's write here that how does the replication takes place in mitochondria. In the mitochondria, again it is having the double-stranded DNA. So here is one DNA and here is the outer strand. So wait, these are the outer and the inner strands. Now this inner strand is also referred to as light chain, and this outer strand is referred to as heavy chain or the heavy strand. So it has its ori and it has its ori. It is known as ori of heavy chain, ori H, and it is having ori of light chain. So this is one thing you need to know. Secondly, here also there is the unidirectional mode of replication, and then main important one here is that there is one another DNA helicase which comes here to do the melting and all. Like as I was talking about the DNA helicases in prokaryotes, those DNA helicases here are not involved. So here there is one important helicase, and if you need to know its name, it is twinkle helicase. So this twinkle helicase is doing the melting and is opening this loop so that the replication can easily begin. So these are the some of the important significant topics which are important to know the perspective of the prokaryotic and eukaryotic replication and to maintain the connecting link between them. Now we should come to the another important part, that is main replication in eukaryotes. Moving on to the eukaryotic replication. See, more or less, if I say that the eukaryotic replication and prokaryotic replication, they have many similarities also. But then there are certain differences, and I'll tell them. So first important thing is that this eukaryotic replication, the DNA is linear and double-stranded. So on the linear and the double-stranded DNA, the eukaryotic replication has to take place. The DNA which the humans have is bDNA. bDNA means the anti-parallel fashion. If here it is 5' to 3', here it is 3' to 5'. So on this the replication has to take place. One of them will synthesize the leading strands, while the other one of them will be synthesizing the lagging strands. Now there are multiple autonomously replicating sequences which are present inside the DNA of humans as well as eukaryotes. Each of them acts as the separate replicon. They act as ori to initiate the replication. So inside one DNA only, I can have multiple ORS like this. So here the replication will be happening separately. Here the replication will be happening separately. Here the replication will be happening separately, and here also. Later on, lastly, at the last moment, all of these strands, whatever so synthesized, will be joined by the DNA ligase and other proteins. So here we have multiple ARS. So this is, let's say, one ARS. So autonomously replicating sequences are present here. Now these ARS also consist of some of the important genes in them, or some of the important sequences which are conserved in them. So let us take a look on only one of this. Suppose this is my one of the ARS, that is autonomously replicating sequence. So it consists of certain conserved sequences. Suppose this is A sequence, this is B1 sequence, this is B2 sequence, and this is B3 sequence. And this complete is acting as one ARS. So ARS consists of certain conserved sequences, and these conserved sequences are the characteristic features of every ARS. Means every ARS will have these sequences, and these sequences have evolutionarily being conserved. Now we should know about that how the replication will take place. The replication will take place with one of the very important helicase, as we were discussing in the case of prokaryotes. There it was DNA B helicase, but in the case of eukaryotes, the replication the replication starts with one of the very important enzymes, and it is MCM helicase. So here, instead of DNA B helicase, we will require the MCM helicase. Now as the cell will proceed from G2 to S phase, there will be certain cyclins and CDKs formed. These will phosphorylate the MCM helicase so that this enzyme can be activated. This process of phosphorylation of the MCM helicase is called as the loading of MCM helicase. As soon as the loading of MCM helicase takes place, it will do its role of unwinding and uncoiling the particular double-stranded DNA. Now there is another important protein. The protein is DNA polymerase alpha. The DNA polymerase alpha is playing here the role of DNA G primase. So its main function is to add the primers. But once it starts to add the primers, there is a difficulty here. Suppose this is the primer 5' to 3'. Now there will be the nucleotides incorporated by these primers in the primers by this DNA polymerase alpha. Now towards the 5' end, the nucleotides which are incorporated are RNTPs, and towards the 3' end, the nucleotides so incorporated are DNTPs. So this is what you need to know about them, and these are recruited by the DNA polymerase alpha. Later on, the another enzymes come like DNA polymerase epsilon and DNA polymerase delta. DNA polymerase epsilon will catalyze the replication of leading strand, while the DNA polymerase delta will catalyze the replication of the lagging strand. Also, the DNA polymerase delta will be catalyzing the replication of leading strand, but to a much less extent. Now I was talking about the MCM helicase. So inside one ARS, the MCM helicase will be recruited. So let's move on to here. So let's say that here this is my one ARS, this is my another ARS. Similarly, here is my one ARS and here is my another ARS. Now onto this ARS, we know that here is our 5' end and here is our 3' end. Similarly, here is our 3' end and here is our 5' end. Now what will happen is that the replication has to take place. Now the replication here in one ARS will be very similar to what the prokaryotic replication was happening. So for the sake of simplicity, we will divide it into two halves. And now the function of MCM helicase starts. The MCM helicase has to recruit the, first of all, it has to open the particular strands. It has to unwind it and create the bubble. So let's say here in dark green color I have recruited the MCM helicase. This is my MCM helicase. Now this MCM helicase consists of two important proteins on it. One of them is CDC45 and one of them is GINS protein. The CDC45 protein helps to recruit the other members, means it will help to assemble all the members required in the machinery. While the GINS protein will activate and enhance, it will mainly act as the enhancer of MCM helicase. So we have GINS protein, we have CDC45, and then we have the MCM helicase itself, and it will cause the opening of the loop. Later on comes the DNA polymerase alpha, and it will do what? It will start to recruit the primers. So let us draw the primers in red color. So here one of the primers will be recruited, and here the primer will be recruited. Similarly here the primer will be recruited, and here the primer can be recruited. Now we have talked about the DNA polymerase delta and DNA polymerase rho. So now DNA polymerase rho and DNA polymerase delta have to function accordingly. Sorry, not DNA polymerase rho, DNA polymerase epsilon. So on this strand, the DNA polymerase epsilon will work and it will catalyze the synthesis of the leading strand here. But here the lagging strand will be formed. Here also the lagging strand will be formed, and here the leading strand will be formed. So the lagging strand will be catalyzed by the DNA polymerase delta, and this is one ARS. Similarly here also in this ARS the similar kind of replication process will happen. Now you should also know that one of the important proteins here which play the role of beta clamp. We talked about this beta clamp in the prokaryotes. So its role is played here by pCNA. pCNA is the one which will hold the strand. It is a homotrimeric protein and it will load itself on the 3' end. So suppose here is the 3' end and here is the 5' end. So here will the pCNA come, and from pCNA the DNA polymerase epsilon will start its functioning. So this is always, and this is you have to learn. Now we will move on further. Let's make it into the linear form so that we can understand it clearly. Now suppose all of this has been made and now the ARS have been united together. Now after the unison, what will happen and how it will be looking like? It will be looking like there are certain primers here, and then here there will be one primer. Similarly the primers here. So let's not confuse this, I'll tell you. See, if you remember, here was our uh, let's say the division, and here also it was the division, and it was the division of one ARS from another. So we already have learned this, that the lagging strands will be synthesized here, the leading here, right? Let's make one more primer here. And here it will be the again the leading strand. Now as these ARS are united, what happens is that this part remains. This particular part is not joined, and here the nick forms. Now this nick has to be sealed, and this nick will be sealed very soon, and I'll tell you how it is being sealed. Also, side by side there are the primers which were formed. Now before this nick is sealed, you also see that in the primers, if you carefully look at the primers, suppose this was your 5 dash to 3 dash, so here it should be 3 dash to 5 dash. Here in the primers were the RNTPs. And now what is the main role is that we have to remove these RNTPs from the primer. So the RNTPs from the primers are removed by two important proteins. One of the important proteins which will remove these RNTPs from the primers include RNase H. It breaks the phosphodiester bond between the two RNTPs so that they separate. Sorry. The another important protein being the FEN1. It will break the phosphodiester bond between the RNTPs and DNTPs. So here also the bond is broken. Here also the bond is broken. Now what happens is that DNA polymerase delta will fill the gaps, basically these gaps. Whatever RNTPs are removed, the gaps would have been created. The gaps are filled by the DNA polymerase delta. And whatever the nicks are present, I told you about the nicks already, like this nick. The nicks are sealed by DNA ligase. So, to conclude, RNTPs have to be removed. After the removing of RNTPs, the gaps will be created. The RNTPs are removed by the RNase H and FEN1 protein. RNase H breaks the phosphodiester bond between two RNTPs. The FEN1 protein breaks the phosphodiester bond between RNTP and DNTP. In this condition, these will be removed, and then the DNA polymerase delta will fill in it with the appropriate DNTPs. And whatever nicks are left, these will be sealed by the DNA ligase. Now all of this happens, but there is one problem. The problem lies here. Now this is our 3' end. The pCNA cannot move to the very 3' end, and here some of the nucleotides remain exposed. Now this will be further, whatever the nucleotides are exposed, the OH group is exposed, and this OH group becomes very reactive. Now this is called as the end replication problem, and we have to solve this end replication problem so that the OH reactive group is not exposed. And by chance, if this OH reactive group is exposed, this can be cut, and if this is cut, one of the important genes can be lost. So we have to solve this end replication problem, and this is facilitated by the telomerase enzyme. The telomerase enzyme itself solves the end replication problem which are faced here by the particular sequences. Now you see this has been inverted, but you can see it for the simplicity. There is the telomere and it has a protruding end with a G-rich repeated sequence. Now this is what has been put extra, and this is exposed outside. Now what happens is that the telomerase comes here, the telomerase enzyme will come here, and the telomerase enzyme has the RNA template. Now with the help of this RNA template, what happens is that the telomerase enzyme actually starts the replication here. Means if you try to understand it carefully, rather than solving its problem, it is all, more or less even increasing its problem. So it is providing a template. The telomerase is providing a template in a way that this further extends. Now after this has extended, you can see this new DNA which has been formed on the 3' end. This is the red colored new DNA. The whole of the template has been added here. Now what happens is this will move on more further, and more of the template will be synthesized at the 3' end only. Now this much has been done, and more nucleotides have been added, all because of the RNA template which was provided by the telomerase enzyme. Now the telomerase enzyme will be removed, and after the removing of the telomerase enzyme, what happens is that this particular part up till here, it is synthesized by the pCNA again, because the complementary can be matched, and this will be further filled in. So the synthesis will take place on the complementary strand, and hence this 3' end will be filled. At least this part will be created. So how is it created? First it is extended further, and then this synthesis takes place. So this extension is done by the telomerase enzyme. So this is very important, and you should know about this also. Later on there is also one process that I think it's not mentioned here, that this particular part, then even if it gets cleaved, that is not an issue. This is part, whatever it was, it remains protected. So here your end replication problem is also solved with the help of these telomerase enzyme. So here your whole process of replication gets over.